helper cells dependent on the nature of the threat, i.e., what kind of pathogen, whether it's a virus, bacteria, fungus, or parasite. Then also to introduce this concept that not only amongst the lymphocyte effector cells, but even amongst the antigen-presenting cells themselves, there are different subsets depending on the nature of the pathogen encountered. And then following up from the last lecture we had on cytokines, to talk about how different groups of cytokines are associated with these particular T cell subsets to respond to these different types of pathogens. And then finally, I'll be talking about this new class of lymphocytes that have been identified in the past 10 or 15 years, which bridge the gap between innate and adaptive immunity, which we call innate lymphoid cells. And these are we recognize now are very important in bridging or communicating between the parenchyma and for driving in this adaptive T cell response. So, as I said, the environment, we encounter many different types of pathogens, whether they're pyogenic bacteria, viruses that can infect cells, fungi, or large parasites like these parasitic worms. And the types of defense responses we need to mount against each one is dependent on the nature of those pathogens themselves. Whether we generate an inflammatory response, we also need to produce particular cytokines to deal with these. The T cells need to express different sets of chemokine receptors to direct them to the various sites where those infections occur. If they're viruses, you need to mount interferons to produce the antiviral effect, and so on. And the first stage to direct this different type of response amongst the T cells comes with the antigen-presenting cell, either the dendritic cell or the macrophage, particularly, because these are the sensors which capture these various pathogens, and depending on the antigen, whether they're lipopolysaccharides, teichoic acid, glycolic acid, complement tag proteins, or whatever, will direct those antigen-presenting cells to express a particular transcriptional program of co-stimulation and cytokines, which are presented to the T helper cells to direct them into these particular functions. Once these naive T cells get all of these required signals from the antigen-presenting cell, they themselves undergo an extensive transcriptional program which changes their various functions and morphology. So as I said, once they are activated by the antigen-presenting cell, they will express particular chemokine receptors to direct them to the particular parts of the body. They'll change the types of they might increase the expression of particular chemokine receptors to make them responsive to chemokines. They will also produce large amounts of particular cytokines. They'll those cytokines can induce the upregulation of the antigen-presenting cell to express more class 1 and class 2 MHC to become more of an efficient antigen presenter. They might increase adhesion molecules to allow for the extravasation of migrating leukocytes into the tissues, and so on. And so when these differentiated, mature, but naive T cells exit the thymus after undergoing thymic education, we classify these essentially as Th0 cells. Th0 to mark on the fact that they are naive T cells. They have a functional T cell receptor, but they haven't yet encountered the actual antigen. Once they enter the lymph nodes or the spleen and see their corresponding antigen presented by the dendritic cell in this case, depending on the nature of that antigen, will drive them into what are called Th1, Th2, or Th17 cells. These are the major, what we know now already as the major functional T helper cell subsets in dealing with most pathogens. There are likely to be more to be discovered in the near future, but these are the ones that have been most characterized at the moment. Th1, Th2, and Th17. Then you also have another subset, the regulatory T cells, which are opposite to these Th1, Th2, Th17s in that they block their responses. So they are more involved with the maintenance of peripheral tolerance, which I'll talk about in another lecture. So as I've said already, the major function of T helper cells is to produce cytokines which can direct all aspects of the immune response. And they also, through their particular cytokines, they will also drive B cells to make antibodies or undergo isotype switching. The other subset are the cytotoxic T cells, whose major role is to directly kill target cells like virus-infected cells or tumor cells, but they are also a major source of cytokines. So as I said, and just to recap, all T cells are characterized by the expression of CD3 antigen, which, if you remember, is the signaling component for the T cell receptor. And of these CD3 cells, most of these express a receptor which is composed of an alpha and a beta chain, heterodimer. There's a small component which is composed of a gamma delta chain. I won't talk much about these, but they seem to, they're more involved in mucosal immunity, not in systemic immunity, which is what we're talking about here. Within the CD3 population, we have the CD4 cells, which are class 2 MHC restricted, and the CD8 cells, which are class 1 restricted. And as I said, within the T helper cell CD4 subset, we have Th1, Th2, and Th17 cells, as well as the T regulatory cells. This group, the TFH, or follicular T helper cells, I will talk about in 2 lectures' time when I talk about T B cell differentiation, because this specific subset is involved in directing help to B cells to make particular antibodies like IgM, IgE, IgA, and so on, as well as high affinity antibodies. So in the lecture today, I will focus mainly on these three subsets. This is just to basically repeat what I've just said. Now, the first subset, the Th1 subset, each of these subsets, Th1, Th2, and Th17, we mark or differentiate by particular signature cytokines they produce. Th1 signature cytokine is interferon gamma. The action of interferon gamma is mainly on macrophages. When macrophages respond to interferon gamma, what happens is that they increase their lysosomal activity. So basically, interferon gamma helps macrophages, once they've taken in or phagocytosed microorganisms, to kill these microorganisms a lot more efficiently and faster. And this is particularly in the case where these macrophages can take in particular bacteria that can survive and escape from the out of the phagolysosome and whatever, like mycobacteria, listeria, and so on. Without interferon gamma there, these pathogens can survive and replicate inside these bacteria. So interferon gamma is important for stimulating macrophages into what we call angry macrophages to directly kill intracellular phagocytosed microorganisms. Another action of interferon gamma is to activate B cells into making particular antibodies associated with things like classical complement fixation, inflammatory responses, opsonization, etc. So the basic underlying effects of these Th1 responses is to induce an inflammatory response, as well as to stimulate this macrophage into dealing with intracellular pathogens. Right? Now, another thing to note about, particularly with the Th1 and the Th2 subset, is that the cytokines interferon gamma produced by Th1 cells blocks cytokine production by Th2 cells, and vice versa. What is the result of this? This means that when the organism, the body, is infected with a particular pathogen like a bacteria, and you drive the formation of a Th1 response by producing cytokines which block the Th2 response, you are skewing the T helper cell response into dealing with this particular pathogen. And conversely, if you are infected with a parasite, which is dealt with by Th2 cells, the cytokines produced by those Th2 cells block the production of the interferon gamma, and so they skew the T helper cell subset towards a Th2 type response. I've spoken about this before, but always remember that there is extensive crosstalk between the responder T helper cell and the antigen presenting cell. And one of the major communication pathways is this ligand receptor complex CD40 and CD40 ligand, which, as I said, the cytokine interferon gamma produced by the CD4 cell induces the macrophage to upregulate its lysosomal killing enzymes, but also this upregulation also requires the engagement of CD40 by CD40 ligand from the T cell. So the upshot of this is that the macrophage requires cytokine signaling as well as CD40 ligand expression to activate into becoming an angry macrophage. Th2 cells, which are blocked by interferon gamma, are associated with adaptive immune responses to parasitic microbes, particularly helminthic worms. And the mechanisms to deal with worms is based on one, IgE and eosinophils, basophils, mast cells. Now, I'm assuming most of you know the role of IgE, what IgE does. Allergic reactions, right? How do you get allergic reactions?
Interacts with the mast cells.
Right. How does it interact with the mast cells?
FC.
The FC receptors. So mast cells, basophils, and mainly mast cells and basophils have the FC receptor to IgE. So IgE, what you know is that IgE binds to allergens like pollen, food proteins, and so on. And then once it gets engaged by the IgE receptor on the mast cells, causes the mast cells to degranulate and release histamine, prostaglandins, eicosanoids, and so on, right? That is an allergic response. But that is not what the biological organism, that is not what this immune response was designed for. This is a product of our modern environment. These allergies are a misdirected immune response because the original role of this response is directed against parasites in the environment that we take in, whether we drink amoebic water, contaminated water, parasitic worms in the food. These are taken in through our mucosal tissues, whether through the GI tract or the respiratory tract. Remember in the first lecture, I said that these mucosal tissues consist of very fine epithelial barriers which prevent the lumen of these various sites from the underlying tissue, right? So in these sort of infections, you don't want a full-blown inflammatory response, a complement-directed or cytotoxic T cell or a TNF response to break down those fine epithelial barriers, which will allow these pathogens to then invade into the underlying tissues. What you want to do rather is to induce vomiting, diarrhea, and expulsion of these pathogens from the body, which is similar to what you get when you have an allergic response. And so the cytokines which induce these particular mechanisms are IL-4, IL-5, and IL-13. And the effects of these cytokines is to stimulate eosinophils, basophils, and also to promote the production of IgE from B cells, also to increase vasoactive amines and also fluid leakage from these mucosal sites to promote the expulsion of these pathogens. They're not completely inflammatory. You do get a certain degree of inflammatory response because in some instances you do need to directly kill some of these pathogens. And for instance, helminthic worms are killed quite efficiently by eosinophils through the granules like major basic protein and cationic protein. This mechanism of immune response is required because these worms are just too big to be phagocytosed by macrophages or whatever. So you need these other forms of killing to deal with these large antigenic threats. Another effect of these cytokines, IL-4, 5, and 13, is to induce this alternative subset of macrophages, which are called M2 macrophages. In the macrophages that I talked about with the Th1 cells, which are induced by interferon gamma and upregulate lysosomal enzymes and so on, the M2 subset is induced by IL-4 and IL-13, these Th2 subset cytokines. When the macrophage is exposed to these cytokines, it produces, instead of these lysosomal enzymes and these inflammatory enzymes, it produces rather IL-10 and TGF-beta, which themselves are essentially anti-inflammatory and, particularly in the case of TGF-beta, promote wound healing. So this particular subset of macrophages is more involved with homeostasis. Remember, I said that the immune response is here not only to deal with pathogenic threats, but also daily homeostatic mechanisms to remove dead or dying tissues and cells during development and during normal breakdown in our bodies. So you want to remove those apoptotic cells without inducing an inflammatory response. And so this is mediated predominantly by these M2 macrophages. And again, just like the Th1, Th2 story, when you have an M2 macrophage being differentiated, the cytokines produced by these cells blocks the activation of the M1 macrophage phenotype. And conversely, when you have the development of an M1 macrophage phenotype, these cytokines block the differentiation of an M2 pathway. So this is how the immune response is directed into whether it's going to be an anti-inflammatory, homeostatic type function or an inflammatory, microbial killing type function. The third pathway, the Th1 cells, the Th17 cells, their signature cytokine is IL-17 and IL-22. IL-17's major function is to stimulate the production and the influx of neutrophils. And this cell subset is mainly functional at mucosal sites. So it prevents the invasion of potentially pathogenic bacteria within the gut from invading the underlying tissues, but also this cytokine, IL-22, is important in the repair and maintenance of the mucosal barrier at these sites, as well as promoting these epithelial cells to make these so-called defensins, these anti-microbial peptides, to maintain a healthy gut flora, a gut microbiota, and prevent more pathogenic bacteria from invading the gut. So, in summary, Th1 and Th17 are essentially inflammatory. They are Th17s to deal with extracellular bacteria or fungi through the action of neutrophils. Th1 cells, signature cytokine is interferon gamma, stimulating macrophages to deal with intracellular phagocytosed bacteria, but also extracellular bacteria as well. Right? Th2 cells, main function, stimulate allergic type responses, IgE, activate eosinophils, basophils, mast cells, production of histamine, and promotion of I don't want to say non-inflammatory because there is a certain degree of inflammation in that, but non-interferon gamma responses. This summarizes here. Th1 signature cytokine is interferon gamma, Th2 4, 5, and 13, 17 and 22, and this basically repeats what I've just said. Same here. Oh, all right. So don't worry too much about this, but the way we identify whether a T cell is a Th1, 2, or 17, or Treg, is either by looking at the cytokines it produces, or what we can do, you remember what I talked about, flow cytometry? So we have a mechanism whereby when we have a collection of cells that have been stimulated by, say, a parasite or whatever, and we want to look at the particular type of T helper cell response in the blood of this patient, what we can do is permeabilize, remove the cells from the blood of this patient, and then permeabilize the membrane of these cells and allow antibodies to particular proteins to get into these lymphocytes. These antibodies are tagged with particular chemical dyes, and then we pass them through a flow cytometer, and in that way we can identify whether we have a predominance of a Th1, Th2, Th17 subset. The antigens that these antibodies are directed against are against these transcription factors: Tbet, GATA3, ROR gamma T, and FOXP3. These transcription factors are what is responsible for directing a Th0 cell to become a Th1, Th2, 17, or whatever. So by identifying, using these fluorescent tagged antibodies to these important transcription factors, we can characterize whether we have a Th1, Th2 response going on. Now, innate lymphoid cells. So far I've talked about this adaptive cell subset, Th1, Th2, and Th17. And so these cells express CD3, they express a T cell receptor, and they originate or they emerge from the thymus. And in this course, we started off learning that we can divide the immune system into innate immune cells, which are have no memory or no ability to adapt, and the adaptive response, which is made up of B cells and T cells. But now we have a gray area. We now identify particular cells which are intermediate between innate and adaptive. These include natural killer T cells, the gamma delta T cells, and this important group, the innate lymphoid cells, of which there are three major groups, ILC1, 2, and 3. These innate lymphoid cells don't have a T cell receptor because they don't seem to require RAG. Remember, RAG1, RAG2 causes VDJ rearrangement. They don't have a functional RAG1 activity, which means they don't have a T cell receptor. Not having a T cell receptor, they are CD3 negative. And yet, of these three groups, of these three subsets, the cytokines produced by each individual subset corresponds to Th1, Th2, and Th17 responses. So we talk about ILC1, which is similar to the Th1 cells, producing interferon gamma, ILC2 producing IL-5 and 13, so stimulates Th2 subsets, and 3 producing IL-17, just like a Th17 cell. The thing about these innate lymphoid cells is that they are not circulating through the blood like the T helper cells. They are essentially localized within tissues. So they seem to, in addition to the antigen presenting cells in directing these particular pathways, these innate lymphoid cells also function in conjunction with those antigen presenting cells through the first production of interferon gamma or IL-4, whatever, to drive the response into a Th1, Th2, or Th17 type response. Natural killer cells are like cousins of these innate lymphoid cells. They derive from a similar progenitor. And while they are have a similar function to cytotoxic T cells, which I'll talk about in the next lecture, their recognition mechanism is different. So again, these natural killer cells are similar to the ILC cells in that they are also intermediates between an innate and an adaptive immune system. That's all. So, and any questions? No. Okay.